So now we will understand about the next saponifiable lipid, which is esters of fatty acid. So the very first saponifiable lipid that we understood is fatty acids, and now we are going to understand about esters of fatty acids. So esters of fatty acids are results from combination of fatty acid with an alcohol. So when fatty acid combines with an alcohol, this leads to formation of esters of fatty acids, because there is an ester bond formation between fatty acid and the alcohol. So the very first type of esters of fatty acid that we need to understand about is triacyl glycerols. Triacyl glycerols are also called as triglycerides. Right, so this is a combination of glycerol, which is an alcohol, and fatty acid. Right, so this is the formula for glycerol. This is a 3 carbon molecule, right, 3 carbon compound, and this is the fatty acid, where this is the carboxyl group, right, and this is R1 is the alkyl group, like R1 is the chain length, right. So we have not mentioned any specific fatty acid here, and so therefore R1 is written. Similarly, this is second fatty acid and this is third fatty acid. So like this, glycerol combines with 3 fatty acids, right, and the hydrogen from glycerol, right, and OH from each fatty acids combines to get liberated as water molecule, and in between there is a bond formation, that is C, that is R C double O R, ester bond is formed. So the structure gets linked by an ester bond, and therefore this is called esters of fatty acids. Right, now this triacyl glycerol is also called triglycerides, right, and this is a very important compound to understand, because see, when you take extra calories, right, that your body does not need, so where is this extra calorie going to go? So this gets stored as triglycerides in fat cells in your body, right, and these that is stored will be later on utilized whenever there would be a need of energy. So the point to understand is that your body, you already every day take carbohydrates and proteins and all, so this triglyceride the body is not going to use on a normal basis, right. So this will remain stored in your body, because this is extra calorie which your body did not need, right, and therefore this is stored in the body to be available as an energy source when your body does not get enough energy from carbohydrates and proteins, or you have stopped taking food, then your body starts utilizing that stored triglycerides as a source of energy. But see, this is extra calories, and hence it has been stored in your body, and if you keep on taking these extra calories, if you keep on continuing taking extra calories, ultimately the stored triglyceride is so much that this can have a high risk of heart disease, right, because heart disease is related to your arteries, and these triglycerides are nothing but fats, and these gets deposited in your body, in your arteries, your artery which carry pure blood, oxygenated blood, which your body needs, which your heart pumps, right. So if this arteries gets choked with such triglycerides, then this becomes a problem, because the blood flow will not be smooth, and therefore there would be risk of heart disease. So higher the triglyceride, this would increase the risk of heart disease, right. Then the second type of esters of fatty acids are waxes. So these are fatty acid esterified to monohydroxyl alcohol. So these have one hydroxyl group, and therefore this is named as monohydroxyl alcohol. And we all know that waxes are harder and less greasy, so it will not become greasy and sticky in your hand, but waxes are hard, right? And these are long chain non-polar lipids, so we all know that we use waxes as candles and as lamps, right? So these waxes are non-polar lipids, these are not going to get dissolved in water. And waxes are synthesized by many plants and animals, right? So many plants and animals synthesize waxes. The best known waxes from animal is beeswax, right? Beeswax is produced by honeybees. Honeybees are well known for the production of honey, but these are also well known for beeswax. So this wax is formed actually in the 8 wax producing glands that is present in the abdominal segment of worker bees. Now we know that honeybees have various sections. There is queen honeybee, there is worker honeybee, right? So worker honeybees are the ones that make the honeycomb, right? And in the abdominal segment of these worker bees, this wax producing gland is present which produces this beeswax, right? And this beeswax is used by the honeybee for the making of the hexagonal honeycomb. So they require this wax to make the hexagonal honeycomb, but this beeswax is being used commercially also. From the honeycomb it has been extracted, and this is used as candles and as carving materials, right? And as moisturizer also. So this wax is that good wax which is obtained from honeybee, right? So this is one example of wax, which is esters of fatty acid. Similarly, plants also use wax as a protective coating, right? So many times when we see leaves, right, leaves have like water droplets over it, but the water does not seep inside. That happens because of the waxy coating present over the leaf, and this is a protective covering so that the leaf remains protected from any kind of damage, right? So plants also have waxy coating. So then we move towards the third esters of fatty acid, which is phosphoglycerides. Now these are esters of only 2 fatty acids, which means that this is formed by only 2 fatty acids, and there would be one phosphoric acid, one phosphate group, and a trifunctional alcohol, right? So this is actually the structure of phosphoglycerides, where you can see this is the glycerol, right? 3 carbon compound, this is the alcohol, then this is attached to 2 fatty acids, right? So this is esters of 2 fatty acids. There is one phosphate molecule, and with it an amino alcohol is attached, right? So on the basis of amino alcohol, that which kind of amino alcohol is present, depending upon that, phosphoglycerides are of 2 types. These are lecithins and cephalins, right? So let us see that what is lecithin. Lecithin contains the amino alcohol choline. So when in this structure the amino alcohol which is present is choline, then it is called lecithin. So this lecithin is also called phosphatidyl cholines, right? Because this is a phosphoglyceride having phosphate group with choline as the amino alcohol. So lecithins has been extracted from soybeans for use as an emulsifying agent in foods. So this can be used as an emulsifier, and this is also found in the phospholipid bilayer that is present in your body. So in the membrane we have phospholipid bilayer. In that phospholipid bilayer also this lecithin is present. One very important and a good example of lecithin is in the venom of cobra, right? So the cobra, we know when it bites, the person dies because the venom of cobra has an enzyme which is called lecithinase. This lecithinase enzyme is present in the venom of cobra, and what this enzyme does, this enzyme can lead to hydrolytic cleavage of the ester bond of an unsaturated fatty acid, right? So this enzyme can cleavage, that is, it can split the ester bond that is present in an unsaturated fatty acid, right? And this leads to the formation of a product which is called lysolecithin. This is a compound which destroys the RBC, and this is the reason that why when cobra bites, this leads to death of the person. So when venom of cobra has enzyme lecithinase, so this will break the ester bond of unsaturated fatty acids in the person, and this will lead to formation of lysolecithin, and this can destroy RBC. So when the RBC gets destroyed, so this leads to death of the person, right? So this is how lecithin is a known compound in venom of cobra, right? And now we move towards the understanding of cephalins. So as I told, depending upon what is the amino alcohol, on that basis phosphoglycerides are classified. So in cephalins, the amino alcohol that is present is serine or ethanol amine, right? So in lecithin it was choline, in cephalins it is serine or ethanol amine, right? So this is the amino alcohol present in cephalins, and this cephalins are present in nerve tissue of brain and spinal cord. So your brain and spinal cord has cephalins, and see, it helps in accelerating the blood clotting. Now this is a very important thing, because blood clotting is very important, because whenever you have injury, blood starts flowing out, and then the body mechanism is such that the blood can stop flowing by itself, right? That is because of the blood clotting mechanism that is present in your body. So the blood clot is formed so that the blood does not flow so much that it does not flow so much that this damages your body, right? So to stop the extra blood flow, cephalins work in accelerating the blood clotting process, right? So the cephalins work with thromboplastin. Thromboplastin is a compound which helps in blood clotting. Whenever you have injury, this thromboplastin rushes towards the site, and therefore thromboplastin along with the cephalins accelerate blood clotting, right? So these two are phosphoglycerides. So this is it about esters of fatty acids. Now we move towards the third type of saponifiable lipids, which is called sphingolipids. So sphingolipids are class of lipids containing the organic aliphatic amino alcohol, which is sphingosine, right? So like lecithin and cephalin, this class of lipids, the sphingolipids, also contains an amino alcohol which is called sphingosine, and hence the name sphingolipids, right? So there are two types, cerebrosides and sphingomyelin. So cerebrosides are mainly glycolipids. By glycolipids we mean that these are lipids that contain sugar, that is these are lipids that are combined with carbohydrates, right? So we know that glyco term is usually used for carbohydrates, sugars, and therefore these are lipids that are combined with carbohydrate. These are also found in nerve myelin sheath. Now you need to know that what is myelin sheath. This is your nerve cell, right? These projections that you see are dendrite. This is the nucleus, and this that you see is the myelin sheath, right? So what is myelin sheath? Actually, this is an insulating layer that is formed around your nerves. Now, what this myelin sheath help you in? This allows the electric impulses to transmit quickly and efficiently along the nerve cells. So whatever impulses has to transmit, myelin sheath allows in the quick transmission of those impulses, right? So myelin sheath is very important, and the formation of myelin sheath is of cerebrocytes, right? And this is a kind of sphingolipids, right? And then we move towards sphingomyelin. These are the only phosphorus containing sphingolipids. So these are sphingolipids that have phosphorus present in them, and this is also present in myelin sheath surrounding nerves, right? Now there is a disease which is called Tay-Sachs disease, right? This disease is caused by the accumulation of sphingolipids in the brain. So when in the brain there is abnormal accumulation of sphingolipids, so much sphingolipids is formed, then this leads to Tay-Sachs disease, right? And this leads to destruction of nerve cells in the brain and spinal cord. So what happens in this disease is that this disease mainly happens in newborn infants, right? This is also a hereditary disease, right? So what will happen, that these newborn infants, they will actually look healthy up to only 3 to 6 months, and after that these infants, they will start showing symptoms like abnormal development of nerves. They will not have a healthy growth like other infants, and they would be they can be easily identified as an infant which does not have proper growth, right? So they will only have normal growth up until 3 to 6 months, and ultimately they have nerve cells damage, and spinal cord also gets damaged, and they have a very fast death also, right? So this is Tay-Sachs disease. Now abnormal sphingolipid metabolism is also called sphingolipidosis, right? So what is sphingolipidosis? It is the abnormal sphingolipid metabolism. So Tay-Sachs disease is a kind of sphingolipidosis, because in Tay-Sachs disease the sphingolipid metabolism is abnormal, right? So this is it about saponifiable lipids. Now we move towards the understanding of the next classification of lipids, which is non-saponifiable lipids. So we know that non-saponifiables are the one that cannot be hydrolyzed under alkaline hydrolysis, right? So these are steroids and fat soluble vitamins. So see, these are compounds that cannot be hydrolyzed, that cannot be broken down. We need to keep this in mind, right? So these are steroids and fat soluble vitamins. Now steroids are cholesterol, bile salts and hormones, right? So and fat soluble vitamins are mainly of 4 types: vitamin A, D, E and K, right? So we are going to understand each of these in a bit more detail. So first, starting with steroids. So not going deep into the structure, this is a 17 carbon atoms molecule that is bonded in 4 fused rings, right? So these are steroids, and steroids, as I told, are of 3 types: cholesterol, bile salts and hormones, right? So cholesterol is a sterol, that is, it is a steroid which is a waxy substance. So cholesterol is a waxy substance which can get deposited in your body as a wax, right? And these are carried through our bloodstream attached to proteins called lipoproteins. So these cannot travel in the bloodstream alone. These takes the help of proteins. These proteins are called lipoproteins. So what are lipoproteins? Lipoproteins are lipids combined with proteins, hence the name lipoproteins. So these cholesterol, they have to attach with these lipoproteins to travel through the bloodstream. Now this is a very important point. There are two types of cholesterol: HDL, high density lipoprotein, and LDL, low density lipoprotein. Right, now this HDL is often given a tag of good cholesterol and LDL is given a tag of bad cholesterol. Right, so let us understand that what is good and bad in this. So this blue symbol represents HDL and yellow represents LDL. Okay, so this is your artery, for example. This is the blood flow. So the red part is your blood, the blue is your HDL, and yellow molecules are LDL. Right, so see, in this artery there is more HDL, the good cholesterol. So what good cholesterol does? It takes the excess cholesterol back to the liver. So the cholesterol that we do not need is taken back to the liver by HDL. Why is it taken back to the liver? Because liver is the place where all of this cholesterol will be broken down into small particles, and then these can be flushed out from our body. Right, so HDL is something good in a way that it is reducing the cholesterol, excess cholesterol from our body. Right, so we need good cholesterol actually in our body. And LDL, low density lipoprotein, is called bad cholesterol because see, in this figure you can see that there is bad cholesterol, LDL more in amount and the HDL is less. Now see what will happen. Well, when HDL is less and LDL is more, so that means that cholesterol is a wax, and this will choke your body. See the arterial line, it is totally choked by yellow color here. Right, which means that bad cholesterol has deposited in your artery, and hence you can see that the blood flow has become very thin. Now when blood flow is not good, it is going to affect your heart. So you are going to have heart problems. So your heart actually pumps blood to provide to your body. Now since the artery has become thin, so your blood will have to pump more faster to provide blood to your body, and in that way you can have heart disease. Right, so this cholesterol actually chokes your arteries. Right, so this is called bad cholesterol. So we need to have good cholesterol in our body. So we need to decide, this actually comes from the food what we eat. So if you eat more of saturated food, saturated fatty acids, right, so this will lead to the formation of bad cholesterol, as these can choke in your body. But if you eat good food, like if you take good vegetable oils, good unsaturated fatty acids, then this will ultimately lead to formation of good cholesterol. So you need to know that this all comes from the food that you eat. So we need to decide what we need to eat, right, so that our body remains healthy. Right, so this can lead to heart disease if LDL concentration increases. So we need to have more of HDL rather than LDL. Right, so this is the main thing about cholesterol. I think all of you must have heard that cholesterol is not good, but we need to know that there is a good cholesterol also and there is a bad cholesterol also. So we need to have more of good cholesterol. Now we come towards the next steroids, which is bile salts. So we all know that in the liver there is a liquid present. This liquid is yellowish green in color. This liquid is called bile. Right, this bile liquid is present in the liver, and this liquid is synthesized from cholesterol itself in the liver. So bile salt is like a product of cholesterol itself, and therefore this is, and cholesterol we know is a kind of steroid, and therefore bile salt is also a steroid because bile salt is formed from cholesterol, right? So therefore bile salt is a cholesterol, and but bile salt is very important for us because bile liquid breaks down large fat globules into smaller ones, right? So the main function of bile liquid, the bile acid in the liver, is to break down the large fat globules into the smaller ones. So as I told that what HDL does, HDL takes all the extra cholesterol to the liver, right? And in the liver we have bile salts. So bile salts will break down those extra cholesterol into smaller ones, which will then be flushed out of our body, and this is actually important for us, right? So bile salt is also kind of cholesterol which is important for us in our body, and this also aid in the digestion of dietary lipids. So whatever lipids we take are also digested by bile salts. So we need to know that we near our on the upper side of our stomach we have liver, and this liver has bile salt which is composed of a cholesterol, and cholesterol is a steroid, and therefore bile salt is also a steroid, right? So now we move towards hormones. Hormones are called chemical messengers, right? So what hormones does, hormones, they actually are released in the bloodstream, and they carry messages to the target cell so that whatever action has to be done can be done. So these are released in one tissue and transported to the other tissue, so that in a way that they carry message to carry out the action. So hormones, because these hormones are synthesized from cholesterol, so hormones are also synthesized from cholesterol, and cholesterol is a steroid, and therefore hormone is also a steroid, right? So therefore okay, we need to understand that there are other hormones also, but the hormones that are synthesized from cholesterol are called steroid hormones, because cholesterol are a kind of steroids. Now steroids hormones are of two types. These are adrenocortical hormones and sex hormones. So whatever sex hormones we have in our body, the male and female sex hormones are steroid hormones, and also there is adrenocortical hormones, right? These are hormones that are also nowadays these hormone is actually made artificially, and this is taken by athletic people to increase their strength also, right? So this is adrenocortical hormone, and sex hormones are a kind of steroid hormones. So I hope that steroid part is clear with you. What is cholesterol is clear with your understanding, right? And why are bile salts important is also, I hope it's clear. So now we move towards the last part, which is fat soluble vitamins. But before that, hormones are actually the ones that communicate with the body by heading towards their target cell to bring about a particular change. So whenever a particular change has to come, so hormone will communicate with your body so that it can reach, so that the particular change can be carried out in the target cell, right? So now we move towards fat soluble vitamins. So fat soluble vitamins are absorbed along with fat in the diet. So as the name suggests, these are vitamins that are fat soluble, which means that they can only be soluble in fat. Now there are other vitamins also, but those are water soluble vitamin, which mean that they cannot be soluble in fat. They are only soluble in water, but these are fat soluble vitamins that are absorbed along with the fat in the diet. So whatever fat we have in our diet, these gets absorbed with that. Fat soluble vitamins come from both plant and animal food, and we also need to know that vitamins are something, these are actually micronutrients that we require in some quantity, right? These are essential because vitamins cannot be made in our body, so it has to be obtained from outside diet, right? So these are essential micronutrients required in small amounts. We require vitamin in very small amount, but it is very important to have vitamin, right? So these are vitamin A, D, E and K. Vitamin A is good for eye vision, right? So any kind of eye problem, if you have, vitamin A could be the reason that there could be vitamin A deficiency. Vitamin D is also called sunshine vitamin because this is only obtained from sunlight, and these are important in your bone functions. Vitamin E is a very important antioxidant. Now antioxidant is something that flushes out bad things from your body, so vitamin E is also important. And vitamin K is important for blood clotting. So we saw that cephalins work in blood clotting. Likewise, vitamin K also work with thromboplastin in blood clotting. So we need to know that these fat soluble vitamins, as these are fats, these can be stored in your body if we take enough vitamins, right? So these are actually fat, so these get stored in your body. So these vitamins, you have a reservoir of this vitamin in your body. So for some time, if you do not take these vitamins, then it's okay, because the vitamins that would be present in your body would be enough for your body to deal with. But water soluble vitamins are something that you need to keep on taking continuously, because you know that water gets flushed out of your body every day. So water soluble vitamins are very important, right? So I hope that this fat soluble vitamin part is also clear with you. So with this, we come to the end of the classification of lipids. Now see, this is a very important thing actually I wanted to show, though this is not a part of your syllabus, but I think whatever we have learnt, we can understand it with this result. This is a lipid profile of a patient, right? So usually when a person has some problem in the body, it is said to them that you take this lipid test, or you take this glucose test for diabetes, lipid test for if you have cholesterol. So this is a lipid profile, a recent lipid profile of a patient, I know, right? And you can see what is given in the lipid profile. See, cholesterol test is done. Now we know what is cholesterol, right? So the cholesterol results shows that it is 295 for the person, right, which is more than borderline. Borderline is 200 to 239, and above 240 is a high cholesterol. So this person is having 295 cholesterol, which means the cholesterol level is very high. This can have a risk of heart disease. Now see triglyceride level. Triglyceride level is actually less than 150, so this is normal. So we also learnt about triglycerides, right? And then we move towards next test. This is HDL cholesterol, which is 56. So when it is more than 6 when it is less than 60, so it is normal, but we need HDL actually. And but if see, if the HDL level is less than 40, then it becomes a problem, right? So we should not have HDL less than 40. At least we should have 40. Now LDL cholesterol, see, again this person is having more LDL cholesterol, which is bad cholesterol. So this means that see, the highest optimal highest cholesterol that is there is more than 160. So when a person is having more than 160 mg per dl of cholesterol, LDL cholesterol, so that means that the person is at higher risk, the cholesterol is high. So LDL cholesterol of this person is more, again there could be a problem of heart disease, right? And VLDL is very low density lipoprotein, right, cholesterol. So that is all normal, but all I wanted to make you understand is in the lipid profile, if you go to take a lipid profile in any lab, these are the normal things that you will find out, and whatever we have learned, this is a summary of that. So that's why I have kept this lipid profile over here. Okay, so that is it for today. With this we come to the end of the lecture, and with this we come to the end of the classification of lipids. But if you have any doubt, please feel free to ask me. Thank you.